Hi, good afternoon. I'm Liang Han. I'm the clinical analyst at the Division of Therapeutic Performance 1, Office of Research and the Standards, Office of Generic Drugs. I will start this session by discussing FDA's general considerations using the alternative B approach in lieu of comparative clinical endpoint B study for suspension-based metered dose inhalers. This presentation has three objectives. First, I will recognize the challenges with conducting the clinical B studies for orally inhaled drug products. Then I will discuss the available tool and the data from FDA-funded research and the external inputs that contributes to our development of alternative B approach. Finally, I will list some recent PSGs for suspension-based MDIs that recommends alternative B approach and discuss their study design considerations. For local acting MDIs and DPIs, FDA traditionally recommends an aggregated weight of evidence to establish B that includes a clinical B study. But we know there are many challenges for the clinical B study. For example, these studies has high variability and the lower sensitivity due to the flat exposure response. As a result, these studies usually requires larger samples and a long study duration. Consider the huge cost and the long time required for conducting these studies. Now FDA considers an alternative B approach in lieu of the clinical B study that includes in vitro, in vivo, and in silico methods. Ideally, the alternative B approach could assess each step of the local drug delivery from pre-actuation to the local deposition. These steps include the formulation and the device characteristics before actuation. And after actuation, the aerosol is formed. So after the aerosol leaves actuate, the droplet size changed due to the propellant or other excipients evaporation. After the aerosol enters airway, how the patient's anatomy and the physiology condition affect the local drug deposition. So after the final drug local deposition, the dissolution and the absorption from the local site. And we now have many in vitro studies, such as realistic APSD, dissolution, and MDIs that can provide the information for each step during the process. But we have limited in vivo study methods and some in silico study methods that provide information on some of these steps. And the alternative B approach was first introduced in the PSG for the solution-based MDIs, like Cuba and the Cuba Radiator. The PSG states that if the test formulation is Q1, Q2, same as the LD formulation, and that the test device is similar to the LD device, the end applicants can take alternative B approach that includes characterization of emitted sprays, morphology imaging comparison, and the realistic APSD dissolution study, and the in silico modeling, and the alternative PKB studies. And after that, the next question is that can we use the same approach for the suspension-based MDIs and DPIs? To answer this question, FDA hosted a 2-day workshop site invited experts from industry and academics. The workshop consisted of the large conference and the small group discussions. At the workshop, FDA presented the thinking on the alternative B approach and received feedback from the external experts. The consensus at the workshop was, in general, the alternative B approach for the solution-based MDI could also be used for the suspension-based MDIs and the DPIs. But some studies, like the realistic APSD dissolution in silico methods, are more useful to establish BEs in other studies. And the particle morphology charcoal block PKB studies are partially useful or for the confirmatory purpose. Evaporation rate and the velocity profile evaluation, pre-actuation characterization of the formulation have limited utility. It should be noted that the alternative B approach is product specific. That means not a single alternative B approach that works for all the products. So after the workshop, the FDA compiled the outcomes from the event and posted the new PSGs for the suspension-based MDIs. All these PSGs take the option-based approach. If the test product, a test formulation is Q1, Q2, the same as the IOD formulation, and the test device is similar to the IOD device. Oh, shoot. That the option one includes that the conventional in vitro B studies, the in vivo PKB studies, and the alternative B approach include additional in vitro B studies, characterization studies, and the charcoal PKB studies, and the in silico studies. If the test formulation is not the Q1, Q2 as the IOD formulation, and the test device is similar to the IOD device, and the end applicants can take option 2 that follows traditional B recommendations that include a clinical study. And the option-based approach was first introduced in the PSGs for generics referencing the VASP aerosphere and the Bradstreet aerosphere. There, FDA approved the suspension-based MDIs indicated for the COPD treatment. Their formulation contains this DSPC and the calcium chloride, and these components contributed to the formation of the phospholipid-based porous particles. FDA posted the 2 PSGs for VASP and the Bradstreet aerosphere in this February, then posted 4 PSGs for other suspension-based MDIs this August. For option 1, the test formulation, as I mentioned before, should be the Q1, Q2 the same as the IOD formulation. The in vitro B study included the conventional in vitro B study and the 2 additional in vitro B studies that are realistic APSD and the dissolution. Comparative characterization studies, such as particle morphology of the emitted dose, the in vivo PKB study, and with and without the charcoal block. And the in silico modeling is optional for the B establishment, and the test device should be similar to the IOD device. As I mentioned before, the alternative B approach is product specific. You can see here is the dissolution study is only recommended for three products with slow dissolving APIs. And the morphology study, particle morphology study, is only recommended for 2 products that can have with the porous structure. For option 2, the test formulation could be Q1, Q2, or not Q1, Q2 to the ILD formulation. The in vitro B studies contains the conventionals at the in vitro B studies, and the comparative characterization study including the particle morphology, and the in vivo PKB study without charcoal block, and include a clinical BE studies. And the in silico modeling is also optional for the establishment. The test device should be similar to the ILD device. And you can see here that the option 2 is the same for all the products. Okay, now I will discuss the study design considerations for each alternative B study. The GDUFA-funded research outcome indicates that there are many factors that affect the APSD study. For example, the Moss-Slotin model size, the inhalation profile, and there's other factors including the filing time point, and the angle of the Moss-Slotin model inserted. And we also noticed that the response to the various study factors is product specific. And in this figure shows the fine particle fraction variability. It's that for the different products, when different size of the Moss-Slotin models were used in the realistic ABSD study. Therefore, we recommend that your method development should consider the Moss-Slotin types and size and inhalation profiles and other factors. This figure shows that the different Moss-Slotin models and the realistic inhalation profiles and the constant flow rate inhalation profiles. We recommend that the realistic APSD can be conducted at the beginning life stage. If the conventional APSD study shows that no difference between the beginning stage and the end stage. And the applicants also can take a bracketing approach. For example, the small and the larger size Moss-Slotin models plus weak and strong inhalation profiles. As long as the combination of the Moss-Slotin models and the inhalation profiles can cover the expected inter-subject variability of the indicated patient population. Alternatively, you can justify your choice by showing good in vivo correlation if you have this data. And the inhalation profiles should be obtained from patient. And we see some submissions that they provide inhalation profiles from the health subject, and we think it may not be appropriate. And the B is based on the PBE analysis of the impact size mass for each Moss-Slotin model and the inhalation profile combination. And the FDA welcomes that alternative statistical approach if this approach is can be scientifically justified. And you can also request a pre-end meeting request to discuss your proposed statistical approach. Post dissolution study, we also found that many factors can affect the dissolution study, especially the particle size of the emitted particles. And we noticed that currently there is no standard methods. And the method development could be product specific. And in this figure at the bottom, you can see that this figure shows a good dissolution study, because this dissolution study can distinguish the different dosage form and the different particle size. And we also acknowledge that we also acknowledge that the need for the dissolution study is product specific. For fast dissolving API products, the dissolution study may not be necessary. And the PSG recommends the dissolution study should be conducted at the beginning of the life stage. And the study should collect the aerosolized dose of similar drug mass between the T and the IS products. And your method should be optimized and validated by modulating your apparatus sample collection and the dose, etc. And your dissolution study must be discriminatory, that can identify the difference in the deposited drug particle size. And the B is based on the comparative analysis of dissolution profiles with inappropriate statistical methods such as F2 analysis. And after inhalation, some OIDPs could be swallowed into the GI contract. If there is a significant GI absorption from GI contract, the conventional PKB study without charcoal plaque cannot distinguish the systemic exposure from the inhalation portion or the swallow portion. Therefore, for these products, we recommend a PKB study with charcoal plaque that can eliminate the GI absorption and obtain the PK profile only from the local lung deposition. And we understand that the charcoal PKB study with or without charcoal plaque have many similarities in the study design, such as how subjects are recommended for the study, minimum number of inhalations, etc. And currently, the only difference actually is the administration of charcoal plaque. But currently, there is no standard for the charcoal dose and the timing of the charcoal administration. So that's therefore that your method for charcoal administration should be justified in your end submission. And the B is based on the 90% CI for the TR ratio for the AOC and the Cmax being between 80 to 125%. Now we think that the role of comparative characterization study provides supportive evidence for establishing B between the test and the IOD products. For example, that the particle morphology can contribute to the meaning that the in vitro study, the drug performance, such as APSD and the dissolution performance. And in this figure, we see that we have 4 drug products with the same amount of the fluticasone. But the morphology, so the imaging study shows that there's different free fluticasone and the fluticasone associated with lactose. And these different association status can be detected by the dissolution study on the right. And we also, again, that the recommendation for the morphology imaging is product specific. Currently, just as I showed before, that currently this study is only recommended for the products with porous structure. The PS recommended 3 batches of T and IS products and tested at the beginning stage of the product. And the imaging in comparisons should be conducted on the deposited particles of the emitted dose. The morphological features of the particles, which may include the agglomeration and characteristics, should be evaluated. A description of sampling collection method should also be provided. So, and at the end of my talk, I prepared 2 questions for you. Question 1, which of the following statement is not true? So we have 3 options, and I wait a little bit and then review the answer. So the correct answer is C. All the studies in the alternative BE approach need to be conducted for BE establishment for most suspension-based MDIs. This statement is not true. Question 2, which of the following statement is true? We also have 3 options. And the correct answer is C. For suspension-based MDIs with limited GI absorptions, charcoal block PKB study may not be necessary. So in summary, the challenges with conducting clinical BE studies could lead to high cost and longer drug development. To address these questions, FDA has explored in vitro, in vivo, and in silico study design through the GDUFA-funded research projects to identify the alternative approaches that can be used to include the clinical BE study. And following the FDA-sponsored workshop on alternative BE approach, FDA has utilized the input received from industry and academic attendees to aid the development of several PSGs for suspension-based MDIs. And this development, the PSGs, present FDA's efforts to expand alternative BE approaches beyond just the solution-based MDIs, and highlight the additional study consideration needed to apply to the suspension-based MDIs and also DPIs. And so I will wrap up my talk and turn to the next speaker, Dr. Ross Olander. Thank you.